What's up, y'all? How's it going? I'm Amir, back in my basement, and we'll be covering the different types of the fungi a little bit in depth and comparing them. All right, let's get right into it. So first you have cryptomycetes, second you have microsporidians, then chytrids or chytrids, zoopagomycetes, mucormycetes, ascomycetes, and basidiomycetes. The easy way to remember them in order is the mnemonic device, which is can my cock zip line many altitudes backwards, and that's the easy way to remember it. And so for starters, you have your cryptomycetes, which not much is known about the cryptomycetes. Some are parasites of protists and other fungi. They have flagellated spores, which is considered to be a very basal thing. It's only the most primitive, well, Dr. Taft doesn't like the word primitive, but it's the most simple, evolutionarily simple organisms in terms of the fungi that have flagellated spores still, because flagellated spores imply a need to have liquid or moisture or water or something for all those flagellated spores to swim through. So that's why cryptomycetes and chytrids are the most basal of them. They are the only ones that have the flagellated spores. And cryptomycetes, there's like 30 species of them, something like that are known. They're unicellular mostly, but again, not much is known. They're heterokaryotic and they have conocytic hyphae. Heterokaryotic and conocytic hyphae are relevant mainly to the plasmogamy stage. All right, moving on to microsporidians. Microsporidians are unicellular parasites of protists and animals. So notice the difference. This is protists and other fungi. Microsporidians are protists and animals. They have a highly reduced mitochondria. These are also very basal. They have short genome strings. The actual length of the genetic code is around 2,000 units is what Dr. Taft had referred to, which in comparison to more complex beings, it's very, very short. And they don't use flagellated sperm, they use spores. They use harpoon-like infecting spores, which kind of go and they shoot it at the host and use that to get in. They consider it also to be very basal. And an example is Nosema ceranae, which is colony collapse disorder. It's the parasite that causes it in honeybees. And these are also heterokaryotic and conocytic hyphae, and have conocytic hyphae. Chytrids are also extremely basal. They're found in many environments, including extremes, like the geothermal vents. And there are decomposers, parasites, or mutualists, so they've got the whole spectrum. They have flagellated spores, and these flagellated spores are called zoospores or zoospores, however you want to pronounce it. And that's why these two groups, cryptomycetes and chytrids, the only two C groups, those are the ones that have the flagellated sperm and thus are quite basal. And these can be symbionts, or they can operate independently. They can be symbionts with plants or anything, or they can just be on their own. And an example is Batrachochytrium dendrobatitis, which is a parasite in amphibians. In the slides, it shows like this, I don't know, passed out frog that's been taken over by that. And these are also heterokaryotic and can have conocytic hyphae. Now moving on to the zoopagomycetes and the mucormycetes. So both of these share the same life cycles, but they are slightly different. The zoopagomycetes are the first instances of wind-dispersed spores. And this is indicative of the transition to land, where you really don't need don't need flagellated spores, and you actually don't want to have flagellated spores. You want to have the wind disperse them. And these are symbionts or parasites. They have either option. They have filamentous hyphae, which is basically something that's common to all of the fungi, I believe, but don't quote me on that. They have non-flagellated wind-dispersed spores, which is a huge improvement. Then they have a zygosporangium. Both of these two groups, mucormycetes and zoopagomycetes, have a zygosporangium, which is a durable structure which stay metabolically dormant and only release when the conditions are good for release. It's kind of like an what's that called? Something with our bacteria that can endophyte, I think is what it was called, that can stay dormant until the conditions are right. And then that way it can help that thing survive. It doesn't have to reproduce and kill off whatever it is. And that generates as a haploid just a haploid sporangia. And these tend to be with animals and fungi and protists, and they are heterokaryotic and have conocytic hyphae. Now, mucormycetes are a little different because many of them are associated with plants, which is different from the zoopagomycetes, which are animals, fungi, and protists. These guys like to stick with plants, and this group includes molds, very important decomposers, and this group also includes mycorrhizae, a lot of mycorrhizae, and all arbuscular mycorrhizae fall under this group. And these also have a zygosporangium, and some of them can shoot spores. And the example we studied in class was the Pilobolus, which shoots its sporangia towards the light. That's unique to the mucormycetes, and these are also heterokaryotic and have conocytic hyphae. Ascomycetes. Ascomycetes live in many habitats. They're also called sac fungi because of the azocarps, or the asci, which are the sacs that hold it. Sac, that's kind of how I remember it. Those two belong together. Never mind. Okay. They produce fruiting bodies called azocarps, which hold the sac-like asci in the sexual stage. This is only in the sexual reproduction. They can asexually reproduce as well. And they have conidia, which is unique to this group. Conidia fuse with the hyphae of mycelium. Conidia kind of behave like spores in a way, but it's just a little different than that. And then they have a dikaryotic stage, which is different than these guys, which are all heterokaryotic. They have dikaryotic, and which means that they have septate hyphae. And examples are cup fungi and morels, and these produce the ascospores. For distribution, that's a specific type of spore. And moving on to our last group, we have basidiomycetes. Some of them are mutualists, some types are ectomycorrhizae. None of them are endomycorrhizae or arbuscular mycorrhizae because all the arbuscular mycorrhizae, also known as endomycorrhizae, are mucormycetes. And others are parasites such as rust and smuts, which are damaging parasites to plants. These are important decomposers of wood, and the reason why is because many of them are specialized to break down lignin, which is what makes wood into wood, and lignin is also found in xylem and phloem, which is very important for your vascular plants. Karyogamy and meiosis occur in the basidium, and it includes a long-lived dikaryotic mycelium. So that dikaryotic stage is especially long with the basidiomycetes, which is one of the things that makes them unique. And they have fruiting bodies called basidiocarps, which house the basidia and make the basidiospores. Well, the basidia are the ones making the basidiospores. And examples are mushrooms, your puffballs, and shelf fungus. And dikaryotic stage, they're dikaryotic obviously, especially with that long-lived dikaryotic mycelium, and they have septate hyphae. So the similarities are cryptomycetes and chytrids have the similarity of having flagellated spores. One of them has, the chytrids has zoospores, so it's a little different, but those are the most basal ones. And the theory is that the chytrids split off from the fungal line evolutionary very early, and that's why they are so simple and close to the cryptomycetes. And these two are mostly unicellular. A lot of the chytrids are also unicellular, or some say that the chytrids are unicellular for the majority of their lives. We didn't study that. Don't let the information on Google confuse you, because I don't think we'll be covering that. Could be wrong though. And all of these, the first 5 groups, all have conocytic hyphae, which means that they also have a heterokaryotic stage. These 3, the cryptomycetes, the microsporidians, and chytrids, follow the general fungal life cycle. The zoopagomycetes and the mucormycetes have their own, and that's because they use the zygosporangia. And the ascomycetes have their own, they have the azocarps, and the basidiomycetes have their own, they have the basidiocarps. These 2, azocarps and basidiocarps, are the only ones that produce fruiting bodies for sexual reproduction. Basidiomycetes, we only studied sexual reproduction. We did not study any asexual stage for that, so I'm not sure if they're able to, or if that was just omitted from the content of the course. And these guys are very, very similar, mucormycetes. And one very major thing to remember from mucormycetes, the endomycorrhizae, aka arbuscular mycorrhizae, are all from that group, and that's a very important thing to remember. And overall, let's see, what else do we have? Pretty much all the groups, except for these guys, except for the fruiting ones, all of them have the potential to be parasites, which is strange, but it's just that all the chytrids and most of the cryptomycetes, or many of the cryptomycetes, are parasites. The chytrids and the microsporidians are almost all parasites because, actually I take that back. the microsporidians are almost all parasites because they are so simple that they really can't host on their own, and that's why they're also specially adapted with those harpoons. Overall, I think that's pretty much all there is to it. These things, the easiest way to remember which groups are similar is just where they line up, because these are in the order of closeness in terms of evolution, and that's the easiest way to remember the special traits that each of these groups has. All right, so good luck on the test tomorrow. See you guys tomorrow.